So you guys can download this on web. You can share this. All that information is there. There's supplemental material, which is very valuable. But what I want to do, if we go to the next slide, is I want to talk about the big take-home messages from me as I look at this data set as 7 years of data that we collected in bee pollen and what it means. And I think it has some important messages. I think it has some important limitations that we have to be honest about. And I think, but it still tells us a story. And I think that story is important. Then Kirsten is going to give the presentation. We're going to share, and she's going to share that presentation with you. So if you want to give that to your beekeeping group or modify it for your beekeeping group, you can do that. And then we'll just conclude again with these major points. And the reason we're doing this is we're following that adage, right? Tell them what you're going to tell them, and tell them what you told them. And so I think in that way, we can really try to focus it, because there's a lot, it's of really dense information in this. That's why it's been so long coming out, because it's just a lot of information, and sorting it out has been a real challenge. And so this is a visual abstract. This is actually in the publication. A lot of publications require you to visually summarize the entire experiment in one picture. And so what we tried to do here is show what this experiment was. We looked at pollen for 218 different pesticides and metabolites, and we looked at 1,000 samples over 6 years, well, seven calendar years, but 6 full years. And we looked at a really wide swath of the United States. We really looked at a lot of different states and over a lot of different years. And so that's important. And we looked, and then we categorized these detections, and we found nearly 3,000 different products and metabolites. By far, most of them were viroicides, followed by insecticides and fungicides. But importantly, we also found that in these field realistic conditions, so when we looked at the field, and you guys are the experts, and you could tell if there was chalk root or if there were queen problems, and we checked for nosema and varroa and viruses, what we could do is we could link 5 different ways of summarizing pesticides to some ill effect by one class or other class or group of pesticides. So for instance, we looked at 5 different categories of exposure. So how many pesticides were there? Was pesticides there or not? Was each pesticide there or not on an individual? How many pesticides were there at the same time? And what was the concentration of all these pesticides in the pollen? And then we tried to do 2 areas of risk or hazard. And so we did that by just looking at the diversity of pesticides that were at a level that met some threshold. And also we looked at how much hazard that whole sample had. And we could link these things to very specific disease outcomes. Colonies that had high levels of fungicide had more queen issues, and they had more disease. And Kirsten will highlight that to you. Next slide, please. There are some really big study strengths. There are things we can be proud about this study. One, it's broad, it crosses many years, it crosses many states. It's semi-random, it's not really random. We have some bias just because of the nature of beekeeping and when things happen. But it isn't certainly just looking at sick bees, like for instance, some of the past pesticide surveys have done. You guys are experts. You collected the samples, so you knew what diseases were and could record them. And we had a wide range of all those. We already had a lot of data on honeybee disease because we were doing the viruses and nosema and stuff. So I think this is a really proud study. This was a very difficult, all those samples, coordinating them all. We should be really proud of the work we did to do this. Next slide, please. Study weaknesses. We did not look at everything. There were things we missed. Like I think it would nearly double our cost, and we spend, I think, $300,000 a year on pesticides. Rachel, you can correct me if I'm wrong, but we spend a lot of money on pesticides, and I think this would have increased the price by 40%, just that one product. So we don't have all the products. We certainly don't look at mixes and all those other things or inert ingredients, and that's impossible. Also, there isn't a way of accounting for sublethal effects yet in these calculations, so we haven't looked at that or synergistic effects. We looked only at pollen. We didn't look at wax or honey. We're looking at wax now, and we'll have a report on that in a couple of years, I think. And remember, this is only a single snapshot in time. We didn't look at colonies after. We're just looking at what we found at the time. So these are definitely weaknesses, but I still think, and I hope you'll agree, that we found some important things as well. And so with that, I'll give it to you, Kirsten.
All right, sounds good. So, sorry, Rachel, are you allowing people in? Do I need to keep hitting the admit? I don't know if you still see those.
Yeah, I'm adding them in, so no worries.
Okay, all right, then I won't worry about them. All right, so as Dennis already gave an excellent introduction, here's the DOI. If you want to download that paper, as he said, we'll make that available to all of you, as well as these slides. And we looked at establishing a baseline for real world exposure over 7 years in the United States. And so these are apiary-wide samples. It's an apiary sample of 3 grams of pollen. It's pooled from 8 hives per apiary. So this is not per colony. This is always, all the data we're talking about is per apiary. And from each hive, we would collect at least 4 cells of fresh pollen. Typically, these were 10 samples per state per year if they were participating, although sometimes due to sampling, you would have a few more in one year and then carry over into the next. Altogether, we sampled from 39 US states and Puerto Rico. And so from 2011 to 2017, we analyzed 1,055 pollen samples from apiaries for 218 pesticide residues and their metabolites. The samples were collected throughout the year from January through December. Obviously, there are some regional differences on when you can open up colonies to collect for pollen. And these were often piggybacked onto apiary inspections. And so if you want to see our actual sampling protocol, you can visit beinformed.org APHIS, and you can download all of that. It's also in our supplement. And so here you can just see you guys, apiary inspectors, collecting fresh pollen. We typically avoided the shiny bee bread that had already been in a colony for a long time and tried to collect the fresh matte colored pollen that had just been freshly stored to really give us a snapshot of the pesticides that the apiary was encountering at that time. And so as Dennis had mentioned, we tried to analyze risk. And now there are many, many different ways of looking at risk. And so for us, we looked, do we find any pesticide residue in the pollen sample at all? That would be our pesticide prevalence. And that's a very sort of just, is there pesticide, is there no pesticide? Then we looked at how many different pesticide residues do we find in an apiary sample? So how many different products is that apiary exposed to? And the reason we look at this pesticide diversity is because as you have more and more different pesticide residues, there's more potential for synergistic interaction. And then the third way we also looked at these samples is how much pesticide in sheer quantity do we detect? And that was measured then in pesticide, in parts per billion, and we're calling that our pesticide concentration. And then lastly, fourth way of doing it was how do we disregard the pesticide noise? So often we find a lot of different pesticide products, but some are there at very low levels, and so they're not an ecological consumption risk. And so using the LD50s for each pesticide residue, we took our threshold as being 0.5% of a honeybee's LD50 if she consumed that pollen continuously during her entire 10-day nursing phase. And this is the equivalent of 50 points in our hazard quotient score. And so we're calling these the relevant pesticides of 50 plus, and how many of these are in a sample. So again, it's a type of measurement of diversity. So the abbreviation for that is 50 plus D for diversity. And then the last way we measured risk is the actual consumption, the actual risk from consumption, what we call our hazard quotient. And I'll walk you through how we calculate that a little bit further on, but that is basically taking the residue that's in the sample and dividing it by that specific pesticide's LD50, and then adding those scores up for all of the different pesticide residues in a single sample. And so as Dennis had mentioned, one of the problems we did have is in sampling. Due to fiscal year funding cycles, sample collections in 2011 started in July. You can see that as the orange ones at the very bottom, left side of this graph, whereas in 2017, our sampling was from January through June. So 2011 and 2017 don't overlap at all. And as of July 2017, due to it being a new fiscal year, we switched over and started taking wax samples to analyze for pesticide residues. So all of these samples are collected from July 2011 through June 2017. And as I said, 1,055 samples were analyzed. And so if we look at pesticide prevalence, just whether or not there are pesticides in the sample at all, you can see that the majority of these states are tending towards dark red being 100% of samples have at least one pesticide residue. And for every state, this is really the mean overall sample years. So it may have varied from year to year, but on average, most of our samples across the entire United States, 80% have some sort of pesticide residue, and about 20% are completely pesticide free. Hawaii is a bit of an outlier here with very low pesticide prevalence. And so here you can see from year 2011 to 2017, we're using the same color scheme throughout. So when we talk about all pesticide residues, those are always shown in gray. When we talk about insecticides, pesticide residues that are insecticide, we always show those in yellow. For fungicide in orange. For herbicide, we chose green. And for varroacides, we chose red. And you can see that over time, the amount of insecticides we're finding is going down, but over time, the number of fungicides and herbicides we are seeing in samples is tending to go up. And so our second measure of risk is pesticide diversity. Here we're showing a map on a scale of 0 to 10. The maximum number of pesticide different products we find in a single sample is 21. And then so the mid-tone here is a 5. And you can see that California and Nebraska are slight outliers in this. They have slightly more concentration over all sample years. In Nebraska, this is often because we find 2 different neonicotinoids in a single sample, whereas in California, we tend to find a lot of fungicide residues that are adding to the pesticide diversity. And so here you can see the number of different residues we're finding. Most samples have somewhere between 1 and 3 different residues. As I said before, we do find up to 21 different products in a single apiary in a single sample. The mean across all samples is 2.78. And if we ignore the 189 samples, the 18% of samples that are pesticide free, then the mean pesticide for ones that are pesticide positive is 3.39 products per sample. What I do find slightly concerning is that we do still find quite a lot of samples with 5+. There's quite 70 with 5 and 42 with 6+ pesticide residues. So there are definitely apiaries that are having a wide exposure to a lot of different pesticide variety. And when you have high pesticide diversity in a single sample, you have much more potential for synergistic interactions. And so when we look at pesticide diversity over time, for all pesticides, that is increasing significantly over time. You can see that in the 2014, from then on, we tend to find more samples that are above those 5+ pesticide residues. When you look by class, you can see that insecticides are actually going down over time, whereas fungicides and herbicides are increasing over time. So the pesticide diversity is mimicking the patterns we saw here with pesticide prevalence. And so when we look all together, we had 2,933 pesticide detections over our 1,055 apiaries. The majority of those detections are varroacides. Over 40% of all pesticides we find in pollen are varroacides. This is not surprising. Varroacides are applied directly into the hive. You would expect some of those to end up in the pollen. The bees are also storing in the colony. And then the next 2 groups are insecticides and fungicides, followed by herbicides. And other could not be classified. So these are things that are like bird deterrents or other ingredients for which we were testing that don't fall into those 4 classes. What I do want to point out is of those 2,933 detections, only 60 of them were neonicotinoids and 62 were insect growth regulators. So there's a lot of media attention to neonicotinoids, but at least in the pollen samples that we're finding inside the colonies, the bees are not frequently storing neonicotinoids. As we'll see as I continue with this talk and we talk about the significant residues, When they show up, they're often high ecological risk to the bees from the consumption of it, but we just don't find them all that often. And so we also looked at pesticide concentration. Now, if you look at pesticide concentration and you don't limit it, we limited this map to a scale of 1,000 because that's the threshold where you just have a high concentration. And if bees have to detoxify even a sublethal pesticide, this could potentially tie up their detoxification machinery if there's a high concentration of it. And so our 1,000 in concentration in parts per billion was our threshold there. And you can see that our ag intense states, places like California, New York, New Jersey, where they have a lot of specialty crops, tend to have high levels of pesticide concentration. The main drivers towards pesticide concentration tend to be our fungicides as well as our thymol residues from varroacide. And again, as I had mentioned previously, these are means over all sample years. If you want more specifics for your state, we do have a table exploring that in greater detail for you for your particular state in our supplemental of this paper. And so when we look at pesticide concentration, it's not changing for all pesticides over time. However, we are finding an increase in insecticides and a decrease in varroacide concentration over the 7-year survey. And so that's in that yellow, in that second bar of this graph, and the bottom one, the varroacides on the very bottom. And so pesticide concentration, if you look at it for our high concentration samples, our samples that are over 1,000 parts per billion, of the 1,055 samples, there were 105 of them. As you can see from this chart, that light pink dominates throughout. And that light pink is from thymol. I'm just pointing this out that beekeepers who are using thymol, we find them very high concentration of thymol in the pollen, most likely because beekeepers have treated recently. It does seem to dissipate out of the colony very quickly, but at the time it's being treated and applied, you do find very, very high concentration, up to 55,000 parts per billion of thymol in a given sample. When you remove thymol out of that, the number of samples that meet this threshold of 1,000 in parts per billion drops to 62. And as you can see, each one of these bars tends to have multiple colors in it. That indicates that multiple different products are adding up to make that 1,000-point threshold. It's not one single driver in most samples that is causing that high level of pesticide concentration. And I just wanted to highlight that because that differs very strongly from what we will see later with the actual consumption risk in the hazard quotient score. And so here you can see that there were fungicides often detected at 1,000 parts per billion. And I just wanted to highlight this because we find that colonies experiencing queen issues often had elevated fungicide HQ scores. Now, fungicides are considered bee safe, and so these scores are still very low. The colonies with queen events had 3.82 that was being added by fungicides, whereas the colonies that were normal and were all queen right had on average 1.18 HQ contributed to fungicide. As I said, fungicides are considered bee safe, and so they really don't add to the hazard quotient score, but we do find them in really, really high parts per billion. Often you have to divide this parts per billion by about 200 because that's the LD50 for fungicides. So that's why they add very little to the score. So how do we actually calculate our hazard quotient risk? Well, this is the consumption risk. So you usually take a dose and you divide that by a screening benchmark. And then so in our case, we take the pollen pesticide residue and we divide that by the LD50 for that particular product. And so that's parts per billion measured per kilogram. And then because a bee during her nursing lifetime consumes about 100 milligrams during that 10-day nursing phase, you end up having a threshold of 10,000. And so normally you would have to divide this parts per billion divided by the LD50 by 10,000. However, your screening benchmark is supposed to be a no adverse effects level. It's not supposed to have a negative impact on the bees. And we're using an LD50. And LD50 is what causes 50% mortality in a cage trial with adult bees. And so obviously that's not a no adverse effects level. And so instead of using the LD50, we have to build in a safety factor. And so we use the EPA of 10%, which is a food safety threshold. So instead of dividing the score by 10,000, you would have to divide the score by 1,000. However, if you divide a score, let's say of 1,000 by 1,000, you end up with a score of 1. Whereas if it's below our threshold of 1,000, you end up with a score of about 0.6. And it's just very difficult to conceptualize risk when numbers are very small. For example, if I ask you to lend me a penny or a dime, you'll all reach in your pockets, you won't think twice about it. However, if I ask you to lend me $100 or $1,000, you might lend me $100, but if I ask for $1,000, you're going to want some sort of guarantee that I can pay you back. And yet the difference between a penny and a dime and 100 and 1,000 is exactly the same factor of 10. However, when scale is small, it's really hard for us to conceptualize large differences. Risk is just easier to understand in larger values. You understand the difference between 500 and 1,000 much better than we conceptualize the difference between 0.5 and 1. And so because those values below are hard to visualize in our minds, we're just going to present our HQ score as the parts per billion divided by the LD50, and we're not dividing by 1,000. And so when you end up with a score of around 1,000, that is 10% of a honeybee's LD50 if she is unable to detoxify this, and this is her only food source during her 10 days of nursing. So if the score is below 1,000, we would expect harmful effects are not likely. If the score is right around 1,000, the contaminant alone is not likely to cause ecological risk. But then you add in other stress factors like varroa or poor nutrition, then scores around 1,000 may be just that straw that breaks the camel's back. And then if you have scores above 1,000, harmful effects cannot be ruled out. And so here's an actual sample where we had 3 different residues. One of them was the breakdown product from Amitraz, DMPF, that was detected at 29 parts per billion. The chlorothalonil, the fungicide, was detected at 111 parts per billion, and chlorpyrifos, I'm sorry, I was reading the LD50s and not the actual parts per billion. But you do the math and you end up with a score of 150. So this is well below our 1,000 point threshold. Here is another sample. Again, it's contaminated with 3 products, but this time it was contaminated with an insecticide, a fungicide, and a neonic. And so when you do the math on these using the LD50s, you end up with a score above 1,000. It's 2,033, and that's predominantly due to that thiamethoxam in the sample detected at 39.6 parts per billion. And so, as I had mentioned before, we were trying to eliminate the noise in the system. So those pesticide residues that are in the pollen but aren't causing any real consumption risk because they're contributing less than 0.5% of a nurse bee's LD50. And so here, this is on a scale from 0 to 1. So if a state tends to be more towards the dark red, like Florida, that means on average in those samples from Florida, we're finding at least 1 residue that is contributing 50 points. 50 points, again, that's 1/20th of our 1,000 score threshold. This is just really to eliminate sort of what we're terming sort of pesticide noise. What are the pesticides that are showing up and really contributing to our HQ scores? And when we do this, beforehand, you saw that we had 2,933 detects. But when we only look at pesticides that are contributing at least 50 points, we now only have 349 detects, and over 90% of those contributors are insecticides. The other 7% are varroicides, whereas herbicides and fungicides, there was only 1 single detect that contributed at least 50 points in either of those classes. Of the fungicide and the herbicide. Now, if we look at the neonicotinoids, there were 45 of those 60 neonicotinoid detects that contributed at least 50 points. And so now this is a much larger piece of the pie. It's now 12.7%, whereas before it was only 2%. And so if we look at the 50+ diversity over time, you can see that it's actually going down over time. in the all pesticide and in the insecticide class, and there just really aren't enough detects in the other classes for you to make any, there's no significant difference from year to year. And so the actual hazard quotient score, this is the actual consumption risk. Our threshold is 1,000 points. 5.4% of all samples are above that threshold. And again, our map is scaled from 0 to 1,000 as it was for parts per billion, except now this is due to ecological consumption risk. And you can see that Oregon and Nebraska and Delaware are the outliers here. Oregon predominantly because a lot of the samples we were looking at in that state had high levels of perlethrin, which is a nuisance pesticide, and then in Nebraska predominantly due to neonicotinoids, which we also find some of in Florida. So when you look at the hazard quotient score over time, it's not changing. It's very erratic over the years as to the different pesticide classes, and so it's not significantly different from year to year. And so remember before I had shown you a very similar graph, but for the concentration, and each one of those bars had a lot of different colors in it. Here we see the 57 samples, the 5.4% of all samples that are contributing at least 1,000 points. And you can see that for most of these samples, the bar is almost entirely one color, meaning that one insecticide is the main driver of the risk to this score. The only difference, the only outlier here is the samples from Nebraska, because often those were contaminated with two different neonicotinoids, thiamethoxam and clothianidin. The one is the metabolite of the other, so we don't know if it was already being broken down in the pollen, and that's why we were finding both, or if different fields that the bees were foraging on, picking up pollen from, had the different neonicotinoids. And so again, all the blues in this one represent the neonicotinoids. And so for our high risk ecological consumption risk samples, neonics, when we find them, do seem to be large contributors to this consumption risk, this HQ score. And these are the insecticides that we really keep finding as big contributors. It's chlorpyrifos, the 3 neonics, bifenthrin, and carbaryl. Oh, and I'm sorry, and the perlethrin is predominantly in Oregon. You can see those last 3 Oregon samples there towards the far right have a lot of perlethrin, and that is also what we were finding in West Virginia. And so here, it's just to emphasize our total pesticide detections. This is again the diversity. I'm just plotting it out so you can see that that red line is where we find 5 or more pesticide residues in a given sample. In a prior study we'd done with commercial beekeepers, when we found more than 5 different pesticide residues, it was linked to a number of health risks such as queen replacement and high pesticide residues. And you can see the means from year to year. So altogether, there are 2,933 pesticide detections. But if we look at only the 50 plus, again, that drops to 349, whereas the max detection in a given sample for all pesticide residues was 21. If we only look at the 50 plus, the relevant detects, the maximum any sample had was 5 different pesticides that were adding at least 50 points each. We also then looked at varroa infestation and different varroicides. Please keep in mind that these results were taken from 2011 until to 2017, and there may be some resistance changes in varroa since then. But on the bottom of this graph, you see the numbers 0, less than 3, less than 5, less than 10, and 10 plus. That is the amount of varroa per 100 bees in that apiary. And you can see that the apiaries that had 0 varroa mites, so they had just been treated, the beekeepers were keeping their varroa mite levels really low through management, they tended to have much higher levels of the amitraz breakdown product, DMPF, and Coumaphos. Whereas the colonies that had very high levels of varroa mites, that's 5 to 10 or 10 plus varroa mites, which you're seeing in the far right of the graph, tended to have high levels of both fluvalinate and thymol. So we don't know if beekeepers are noticing that they have high varroa mites and they don't know how to get those down, and they're putting fluvalinate and thymol in to try and bring them down, or if those products just weren't helping to bring the numbers down. Again, these are just correlations. These are not causations. And then we also looked at nosema. Altogether, we sampled 1,034 apiaries of our 1,055 for nosema throughout the apiary-wide sample. Now, this is not per hive, but just any one of those colonies in that apiary could have been nosema positive. On average, we found that 43% of our apiaries had nosema, and on average, it was 0.3 million spores per bee. The apiaries with undetectable nosema had on average, and this is in row B so it's the second row in this very complicated chart, they exhibited 20% lower pesticide diversity compared to the apiaries with nosema. So let me walk you through this graph real quick. On the bottom, you see the 0, the less than 1 million, and the 1 million plus spores. The gray is the representation for all pesticide detections, whereas the orange is just for fungicides. And so you can see that there's no difference in prevalence in the top row between colonies that had no nosema with the colonies that had nosema. However, in that second row labeled diversity, this is the number of different pesticide residues. And we see that colonies that had no nosema tended to have less pesticides overall, and they had significantly fewer fungicides than colonies that had the million plus or somewhere between 0 and 1 million spores. If you go to concentration, there was no significant difference for all pesticides, but the colonies that had high levels of fungicide concentration also tended to have nosema. You can see that in the two, so the middle and the far right in that C bar, those orange bars are higher than the one on the far left. And the same pattern is true for the HQ score. Samples with nosema on average from the fungicide HQ, and because fungicides contribute very little, you have to look on the right side to see the scale on that. That goes from 0 to 2 to 4. And so colonies with nosema tended to have on average a fungicide HQ score that was 3 times higher than colonies without nosema. So it looks like if colonies are exposed to a lot of fungicides, they also tend to have nosema. Again, these are correlations, not causation. It is interesting since nosema is considered a fungal spore. This may be that it's just getting rid of the good fungi in the colony when you have high levels of fungicides, and thus allowing the bad to proliferate. Sorry about that. And so then I just wanted to break down and sort of draw attention to the pesticides that were showing up frequently in the 50 plus. So these are the ones that show up in the what we're calling relevant pesticides that add at least 50 plus score points, HQ points in 10 or more samples. And you can see that they're all insecticides except for fluvalinate, and a lot of the neonics show up here. So clothianidin, imidacloprid, and thiamethoxam all show up. And I wanted to highlight the ones that on average are contributing quite a lot to the HQ score. So carbaryl on average is contributing 2,500 points. So that's above our 1,000 threshold. Clothianidin is the same. And prolethrine is really the outlier here. When we find prolethrine, it's often at a very high score. The max it ended up adding to any individual sample was 29,000 points. So that's, of course, almost 30 times above our threshold, and that is of concern. Prolethrine has been used for treating both mosquitoes and nuisance pests, and so it's often used as a wasp and hornet killer. So I don't know how the bees are picking this up and bringing it home in their pollen, but when we find it, it is problematic. And so we also looked at brood disease. Now, not all of the samples that were returned for pesticide analysis also had those data sheets that we hand out to apiary inspectors, but 14.3% of all the samples we analyzed for pesticides also had these apiary inspection sheets. And so they were inspected for overt symptoms of honeybee diseases. We looked for American foul brood, European foul brood, sac brood, chalk brood, and snot brood. Brood disease was detected in about 30% of those inspected apiaries. So of the 151, 45 of them had brood disease. And we found that the HQ for fungicide was elevated in samples that were positive for brood disease. So the colonies that tended to have brood disease, their mean HQ fungicide score was 4 versus 1.2 for the colonies that were free of brood disease. Again, that just seems to suggest that when we end up with a lot of fungicides in these colonies, that seems to be a high stress factor, and we're ending up with nosema and these brood diseases. So in conclusion, the summary is not all pollen is contaminated. Overall, 18% of our apiary-wide samples were pesticide-free. So 18% of apiaries are not having any pesticide residue at all, not even from varroacide in the pollen we're collecting. Only 5.4% above were above our HQ threshold of 1,000, where we might start to see some sublethal impacts on colony health. The pesticide diversity is potentially increasing over time. So although samples were contaminated with many pesticides, as I had said, we found a max of 21 different pesticide residues in a single sample. On average, each sample we collected had over 2, on the way to 3, 2.74. However, most of the time, a lot of these pesticide residues we're finding are not contributing significantly to the consumption risk from that pollen. The max we find that at least 50 plus points in a given sample was 5, and on average 0.3. So every third sample we're looking at on average has at least one pesticide residue adding 50 plus points. Regarding the pesticide class, the majority of the hazard quotient score is contributed by insecticides. That's not surprising. Insecticides are designed to kill insects. Honeybees are insects, so these tend to pose the highest ecological consumption risk to honeybees. Neonicotinoids were not frequently detected, but when we did find them, they dramatically increased those HQ scores. And that you could see that those blue bars were very, very prevalent in those 57 samples that were above our 1,000 HQ threshold. Also, over time, fungicide residues are increasing. The quantity detected went from 4 that we were finding in 2007 as a max in our sample up to a high of 10 in 2017. And although fungicides are not a large contributor to HQ because fungicides are predominantly considered bee safe, we are often finding them at a very high part per billion. So they contribute a lot to pesticide concentration, and that may just be stressing out the health of the colony because they still need to detoxify these foreign substances. And the fungicide residues are tied to both an increase in brood diseases and nosema. They were correlated. When we find high fungicide loads, we also tend to find brood diseases and nosema. And so I'm going to hand this back to Dennis. Oh, Dennis, are you there?
Yep, I'm right here. Yeah, thanks, Kirsten. Thanks for that big thing. So there's a lot of information there, and I think you'll agree that if you just step back and I listen to what I've heard, it is that I think for one of the first times ever, we really demonstrate that pesticide in different amounts that we see in the field have stress outcomes we can measure in the colony. However, I think it's safe to say that if you exclude synergistic effects that might be real and we haven't captured, that most beekeepers are probably not being affected by a lot of negative effects from pesticides. That said, there are some beekeepers who are, that there's clearly a lot more pesticides there should be in some samples. And I think this study also helps highlight which pesticides we should probably go after, because it looks like perhaps someone's misapplying it, or there may be other things to try to figure out why they're there. So I think that's my big take home. And I think we'll just we'll take questions from there. So thanks for your attention, and hopefully that helps you understand this important body of work that a lot of you guys in the apiary inspectors and the Informed Partnership and tech teams and all have helped create this really important, I think, database.
Thank you so much, Kirsten and Dennis. That was great. Thank you. If you would like to ask a question, feel free to unmute yourself and ask now, or you can drop your question into the chat box and I will ask it for you.
Hi, Blaine White, Minnesota here. Question regarding thymol and pollen. Did you notice any geographical areas? We have some wildflowers that honeybees work here in the summer, the genus Monarda, that the stems of the plants after they're dried have a very high thymol levels. And I'm wondering if pollen could be a source of some of those high thymol levels that you're finding.
Yeah, certainly that's a possibility. We're going to do some more analysis. We're working with someone who's going to look at to see if there are clusters with some of this stuff. Certainly that's a possibility. I think we're seeing at really, really high levels, though. It would be awfully surprising if I think you'd smell that plant if you walk by, if the pollen had that much. Like we're seeing a lot. So it would be surprising if that was natural, but certainly it could contribute to perhaps the prevalence we see.
The dried plants literally smell like apigard thymol.
Okay, well, I don't know.
These are they're very high levels in the plant, so
okay, so
that's why I'm asking.
You think the bees are collecting enough to make it smell? So then, yeah, sure, we can look to see if there's a cluster. So I don't know. I would wonder what the evolutionary advantage of a bee producing thymol in its pollen would be. I can see it in the stem because you'd want to protect against herbation, but you wouldn't want to scare bees away. So it's interesting. I've always been surprised, but it's possible. And I guess that might help point that out. Kirsten, you want to say something?
Yeah, I mean, you'd have to look at the pollen from Monarda. I mean, bee balm is definitely visited frequently by bees and butterflies. I wasn't aware that there was thymol in the plant. So I can't comment one way or the other. As far as I know, that when we looked at it, there wasn't any geographic cluster from thymol. And I know from some of the Sentinel apiary data is when they treated with thymol, we saw high spikes in the pollen sample, the pesticide residues afterwards. So I don't know if plants are contributing. That may well be. We're definitely finding very high levels of thymol in a lot of samples.
Okay, I have a question in the chat. Danielle Downey asks, were the demographics of the beekeepers analyzed?
No. There was a lot here. This paper is so dense that to squeeze something else out of it would have made us, we wouldn't have finished, like it just, yeah. No, this is a dense paper. There's a lot more follow-up work we can do on these pesticide, but I think that this is as dense as you'd want a paper to be.
It's a little denser than I wanted it to be.
Yes, it's much denser. We just couldn't figure out where to stop or where to start. Yeah.
Kathleen Prowl asks, any certain fungicide is being found? So was there any fungicide that you found repeatedly nationwide?
So you can look at that up. That's all in the supplemental. It's all written out. So you look that up. I wouldn't have remembered it. I doubt Kirsten, you would remember it. We'd look it up too. So it's all out.
No, I would check that table in the supplement. It tells you all of the fungicides, how often they were detected, how often they are above the 1,000. I can tell you real quick, the high ones in fungicides, we tended to find a lot of chlorothalonil and a lot of THPI. Those were the two I would be most concerned about.
Joan from New York asked, will this presentation be located for further viewing and sharing? Yes, we will be posting that onto the National Honeybee Survey website, and I'm gonna put that link into the chat here. And then also I have another question from Danielle Downey. She said, in Hawaii, there were consistent high detections of weird chemicals. Any success stories about tracing back those exposures?
No, I mean, that is the shortcoming of the National Honeybee Disease Survey, right? Like you guys all have pulled out hair and screamed at us because the results come a year later than the collection, right? Because it's not met real time. So I think there's just too much delay between what we find and detection. We've tried to model that with the Sentinel apiary, where we try to get a rapid turnaround with the results from Sentinel so that we can right away communicate with farmers to try to figure out why we've seen weird spikes, but we can't get an analysis fast enough to make that practical yet. So it's a really good idea. That's the ultimate goal, right? Is to say, not to be confrontational with farmers, but to say, hey, we noticed this spike here. What do you think that might have been? Because maybe some of this is just the bag is open and the bees think it's pollen and digging it out, right? So there might be obvious things to do, but it's sort of like the COVID. You have to have rapid tests for it to work, and we don't have those rapid tests.
And then I have a question from Keith Tignor in Virginia. It looks like concentrations were higher in certain states. Any thoughts on this occurrence and why that is?
So yeah, actually, there was no variance across the states. I had limited that map to our 1,000 point threshold to highlight the states where on average it was about above 1,000. Normally that map would have gone up to 3,000. And when you do that, there is no statistically significant difference between states in concentration. There do seem to be some states that on average are above the 1,000, and we list those in the paper, which states those are. I think there's about 5 of them.
Kirsten, if I could follow up on that, this is Keith.
Sure.
It looked like in your graph of the concentrations, the PDB it included paradichlorobenzene. Was I misreading that graphic?
No, it's parts per billion is what it was in parts per billion. I think we looked at paradichlorobenzene as one of those residues, but it's at 218, and I can't remember off the top of my head. This paper has been a long time since I actually worked with the data, so okay. I'd have to double check, but if so, it's in that table.
Any thoughts on why, given that we're supposed to be having low exposure to paradichlorobenzene, if it's used for moth control, any thoughts on why that would have been higher? Like I said, it looked like in certain states it was higher than others.
We definitely didn't have a map where we were just looking at paradichlorobenzene. The map should have said in parts per billion, so PBB, parts per billion, PPB.
I mean, I'm trying to think, did we find a lot of, I don't even know that we found, I mean.
I'd have to go back to the raw data.
I think you're reading what we're meaning as a measurement, which is parts per billion.
This is Blaine. It sure looked like paradichlorobenzene was on the list when the slide was up.
Okay.
It's not surprising. It's used for wax moth control.
Right.
So paradichlorobenzene was only detected 31 times total in all samples.
But you have you can look at the paper and that helps clarify it maybe.
Yeah. I have a question from Tom Steger. Were data collected on land use surrounding the study apiaries that may facilitate some analysis of cropping patterns?
Yeah. Yep. It's a dense paper, Tom. Yeah.
And then Tom also asked, is the basic study design intended to be as random as possible, or are there targeted samples that may represent relatively specific routes of exposure?
This is generally, it's meant to be random. This group, we have moved into a sampling period, as you guys know, now with pollen, where we're trying to take some samples at the beginning of the season and the same apiaries again at the end of the season. So paired samples. This is meant to be random, so no, we're not. Obviously, there is some clustering, like in California almonds, just because it's a lot easier to do do a lot of states and a lot of breadth in a short period of time. But it's generally tried to be as random as possible. There's a clustering in the fall, right? But
so I did just look back at the concentration slide you were talking about, and it's the Nebraska samples that had a lot of paradichlorobenzene. I have no idea why, but the Nebraska samples there had a lot of that hot pink, which was the paradechlorobenzene for adding to concentration.
And I also have a question from Danielle Downey. She says, Judy Wu Smart is studying some very startling point sources of neonics. Could that hotspot be related to seed treatment sites?
It most certainly can. The neonics tended to be predominantly in Nebraska, which is a corn-heavy state, and so it could definitely have been from planting or open seed left out. That I have no idea how they're getting it. We just know they're getting it.
Hi, this is Judy with Smart. I sent in some of those samples, and I do have to say some of those samples were taken from apiaries near Need, Nebraska, or in Need, Nebraska, and I just put a link on a Guardian article talking about this issue, and it's just related to an ethanol plant that is receiving, well, they self-tout 98% of all North American outdated and treated pesticide seed. I don't know how much that really is, but it is going through this plant and resulting in byproduct waste, liquid and solid, with really high fungicide and neonicotinoid levels. And like Dennis mentioned before, there's this delay. So some of these samples were taken there. Some of the colonies died out before we could finish. So some of these samples did contribute to the results, which is probably why you're seeing such high neonicotinoids in Nebraska.
Yeah, we should just touch base with you then, Judy, as we finish that GIS stuff.
Yeah, and it's only been this summer that we've been able to identify what potentially could be contributing to these contamination issues we're seeing in our hives. So this was kind of a newly development, from this year. So I'd love to touch base with you to kind of figure out how we can quantify some of this hazard risk a little bit more thoroughly for our issues.
Yeah, no, I'm happy to talk. We're happy to talk.
Also have a question from Kim Scrim, and I hope I said your name correctly, from Massachusetts. Can you talk about off-label illegal miticide used by beekeepers and its impact on the data collected and analyzed in this paper?
Yeah, but am I going to? No. I mean, we all know, no, I'm not going to say anything. I mean, all the products we tested were legal products. We don't know about if they were illegal or not legal. You guys know the difference between that. I think beekeepers are in a hard spot, right? You have to keep your colonies alive. If there's no choice, they have to do something. So I'm being coy, Kim, and I'm being coy on purpose because we're just trying to show the data. I think if the data shows anything, though, I thought that was a great sign, right? Like if you look at that varroicide, the concentration of varroicides has gone way, way down. Beekeepers are becoming much more responsible. We're not finding so much concentration of varroa. That's one of the only significant trends we found was the decrease in concentration of varroa. So I think it's positive. I think it's positive. Kirsten, maybe you want to be more. You want to talk about it.
I mean, we can just talk about the concentrations we're finding. We have no idea how beekeepers are putting them into a given sample that we get. The residues from licensed products and other versions of the same product, you can't tell in a residue analysis.
Thank you. Oh, wait. Okay, one more question. From Shonda King, do you have any theories as to why perlathrin, sorry, I can't say that very well, is so high in some samples or where the bees are getting exposed to this compound? I did see that in my state, West Virginia, had several samples with very high levels of perlathrin.
Yeah, perlathrin. We actually don't know specifically how the bees are getting into it. It has been used for mosquito control, and I apologize, my other phone's ringing in the background. It is also used for nuisance pests, and so it's used a lot to spray hornets and wasp nests. The amounts we're seeing, it may just be that bees are getting into homeowners' sheds and into chemicals they shouldn't be. I really don't know. But it is used for mosquito control and nuisance pests.
Yeah, I wondered if someone had just sprayed the colony just before trying to get rid of the nuisance hive. There was one amount I was going, wow, yeah.
Yeah.
This is Blaine White again. A comment that mosquito control may well be a major route. We're seeing some up here where individuals are hiring companies to apply materials for mosquito control. We've had some bee kills associated with that when they use encapsulated products, mostly pyrethroids, but they're really hot on the bees. And so something like that could account for it.
Yeah, and when the years that we saw it pop up in Oregon was also the years where they were having some West Nile issues. So it may
Okay.
be due to mosquito control, but it's not a very common, there are other products that most mosquito companies use instead first.
Hey, Blaine, Mike Hansen in Michigan, when that topic came up here in Michigan, especially around the Saginaw Bay area where we have mosquito control districts, our beekeepers were very protective of the mosquito control districts because they found them being extremely careful about the timing of their applications, using more larval products and less adulticides. They are very concerned about this new industry called "I'll spray your backyard for your graduation party," which takes into account nothing about what is blooming and what is happening. It's just, we'll come fog you out so you can have a happy party.
Mhm.
So we want to be cognizant that the major mosquito control players are working very diligently, and then this whole other industry coming along is really changing the landscape on how we feel about that.
Yeah, I would say that the mosquito companies that target homeowner backyards are probably a lot more frightening than the state spray programs that are very well regulated and very, very carefully monitored.
This is Emily Wine, Delaware. I'm concerned that my state was in your top 3 hazard quotient states. And I noticed on your chart, it looked like it was largely due to uses of carbaryl. And I don't know if you'd be able to tease this out, but were you able to get a sense of whether some states were doing a better job of finding softer, lower risk alternatives to some of the high risk pesticides, or why do you think that, for example, the carbaryl levels would be so high in Delaware?
Yeah, so in Delaware, you actually only had 2 samples that had carbaryl, but one of them had an obscene amount. I don't know why.
I also think that, you're hinting on an important thing, and that is, I think we should reasonably expect that beekeeping in Delaware is probably very different than beekeeping in Nebraska than in North Dakota. And so I imagine, I can buy, isn't carbaryl one of those things homeowners can buy and get rid of stuff? Like, isn't that a common homeowner thing too? So I wonder if that's
Yep.
part of it. Some of
It's
it.
basically 7.
Yeah, that's what I
Yeah.
thought. So, I think it's because people can go buy 7 at Home Hardware and it says, put a shake on your rose bush. So they put 7. I think things like that may also be contributing here. I think some of this is just misuse. We're not sure who's misusing it.
So your graph of top hazard quotient states were over a period of, it was over the entire 7 years of the study? Is that correct?
Yes, but not all states were sampled over all 7 years. That's unfortunately part of the problem in this. And so it's the mean for all the samples we have for your state, but it doesn't mean that it was sampled throughout all years. Certain states like California are always sampled, and then other states, they may have only had 10 to 20 samples over the entire study period. It really depended on whether or not we could get someone to take the samples and send them in. So it's not not every state has an equal number of samples. That's part of the problem in the way this was piggybacked on top of inspections.
Okay, yeah, I'm aware that Delaware missed quite a few years of sampling. So do you think that with those hazard quotient numbers, you're actually able to say which states are have higher hazards than others, or are you not able to be confident of that with a lot of states?
I'm sorry, I missed part of the question.
So I know that Delaware did miss quite a few years of sampling. So I was wondering, does that mean that you would not be confident of the hazard quotient numbers?
I would say that when you have fewer samples over the lifetime of the survey, then if you have one outlier, these are means, so it can swing things quite a bit. And so it may just be that you had one or two samples that were really high risk and the others aren't. We do, I believe, have a table, and I can't remember, Dennis, in the supplement where we talk about ranges in the state,
Yeah.
and that'll give you a better sense of
Right.
But where it fell.
But Kirsten, I don't think we found a significant difference between states in any of these things on average.
Not on HQ, no.
No, so there is no significant difference between states, but we list the states that on average had the highest. And I think that's a typical way of presenting data. You sort of present the range and then sort of the high range in terms of risk. So there was no statistical difference, but in terms of the hazard quotient, as we calculated them with the data we had, that was one of the top ones.
Thank you.
The maps were also based on our threshold, just to show, where you're falling relevant to that threshold, to help illustrate it. But as Dennis said, there was no difference from state to state on those HQ scores.
I just want to say, I'm always amazed how much information you're able to drag out of this 45 minutes I spent in a bee yard collecting a sample and putting that everything else together. It's pretty amazing. And if you could have even spent 2 hours explaining all the limitations to the data, we still wouldn't have all of the nuances of this in our mindset as we're finding. Some states sampled some years, some not. But I really appreciate the work and offering this to us today.
Yeah, and this was really designed to help establish a baseline so that we really didn't know what we would find across the US. So this is really, it attempted as best as possible to be a nationwide survey, just so we even know how contaminated are things across the US, what should we be looking for. This is really just meant as a starting point to sort of help aid future questions.
And thanks for saying so, Mike. Yes, we really appreciate you guys doing your 45 minutes. It's a challenge, but we enjoy it. I think as a group, we enjoy the challenge of it all. So thank you all too. I think.
Yes, thank you so much. Before we go, does anyone have a last minute question? And if you think of a question later, feel free to reach out to me with your question and I'll refer that on to Kirsten and Dennis. We are happy to talk about this paper anytime. So if you look at the paper again and have any questions, feel free to reach out to us. And thank you so much for joining us today.
The camera wave. See you guys. Thank you.